Hello and welcome to this presentation of what a drug profile will give you. In our earlier two screencasts, we have showcased which search parameters are available to you and how to do a drug search. Here we will show how our dynamic report generator lets you with ease and speed generate HTML reports directly in your web browser, whether it's a single drug profile or a report with many drugs. As you will see, BioSeeker's Oncology Drug Pipeline Update tool links out to several related internet resources throughout the drug profile. In this presentation, we summarize what a typical drug profile will look like for a particular drug, in this case imatinib Gleevec from Novartis. Now, the imatinib profile starts with listing drug name, drug status, any drug synonyms, active linkage for further searches of imatinib in, for example, PubMed or Google. The drug profile contains active linking to the original investigator or any R&D partners of the drug. Each drug profile starts with a brief description of the drug, followed by filings and approval information of the drug. This particular sections also contains any orphan drug designations of the drug. Our compound information sections gives you compound type, chemical name, CIS number, which links out to chemical depository sites for with chemical structures for further information. We also include basic pattern coverage for the compound as well. Now, the target information is one section which sets BioSeeker apart from what reporter you can have from many other commercial sources available today. First, we start with a brief introduction to what targets the drug in question has. Each target is being described by its official gene symbol with an active link out to the entrance database for further genomic information. We report on chromosome localization, gene name, protein name, molecular function. Then we follow with where in the cells these targets are located, either as a primary or alternate location for the target. BioSeeker has matched all drug targets for target expression profiles in the Human Protein Atlas project, which investigates the expression profiles of targets in 48 different normal tissue types, 20 different kinds of cancer, 47 cell lines, and 12 samples of primary blood cells. BioSeeker gives you direct access to the expression profiles of each drug targets in 20 different tumor types. We report with percentage of strong, moderate, weak, negative, and total number of patients reported for each of these tumor types. And by pressing on the tumor link, you go out to the original source and you can investigate which are the strong expressions of the ABL1 gene, as in this case, which are the moderate ones, which are the weak ones, which are the negative ones, for detailed information. All targets are cross-referenced with a catalog of somatic mutation in cancer called COSMIC. It is designed to store and display somatic mutation information and related details and contains information relating to human cancers. We present you whether or not it is if a target is present in COSMIC, if there has been any recorded mutations within that gene, and if you press on the number of mutations, you can actually analyze in detail what those mutations are, either by site in the gene or by a tumor source, mutational type. Furthermore, we also report which of these targets are also present within cancer gene census, which is a short list of targets which has a strong causal link to cancer, either through germline mutations or somatic mutations. The identity of available biological structures on targets has been retrieved from the RCSB protein data bank for you to easily review what available structures of drug targets exists. For example, for the ABL1, 36 different kinds of structures have been retrieved, where a number of them shows an interaction between the ABL kinase domain together with imatinib. Most drugs have been described with a mechanism of action, including a summary statement and a list of mechanisms. All drugs have furthermore been cross-referenced for the involvement in cellular pathways according to BioCarta, KEGG, NetPath, and NCI Nature. Here we integrate all pathways that include the targets that imatinib is inhibiting. All pathways listed are linked out to detailed information on each pathway. For example, by clicking on the chronic myeloid leukemia pathway, we can find out how imatinib today is suppressing CML through a detailed pathway representation. Under developmental projects, BioSeeker lists every industry-related developmental project of imatinib by indication, highest stage of development, and status. This is followed with a detailed presentation of developmental information by stage of development, starting with phase 3, any sub-indication of cancer, phase 2, phase 1, and preclinical information. Now, you have just finished listening to what information you can expect to find in a BioSeeker drug profile. Thank you for watching.